Hello everyone. So today we are going to learn about the techniques of selection of hybrid cells. But before that, as you know that we are starting our online classes on plant tissue culture. So for details, you can contact on number 9823126925. You can also WhatsApp us on this number so that you can get the details more regarding the classes, and the email address you can also reach to us through email address, that is mailme.bhagyashree11@gmail.com. And one more good news for you guys, that we have started our telegram channel named Roadmap. So you can get the discussion forums are available, and also you can get the notes available through this channel. So if you're interested to get more benefits and more outcome, then you can join this channel. Now, coming to the techniques of selection of hybrid cells, you know that we are studying the protoplast fusion, and we have studied some of the basic subtopics in that how the mechanism is carried out, and which are how the treatment is done, which are the methods that are involved in the treatment. So now there are some techniques for the selection of hybrid cells. Now let's look into detail about this subtopic of the protoplast fusion. Now, selection of the hybrid cells. Now, it is done on the basis of different methods or techniques that are involved. And about 20 to 25% of protoplasts are involved in fusion. But what happens after fusion, that it consists of a heterogeneous mixture of confused chloroplasts. This means that the homokaryons and heterokaryons both are contained. So it is necessary to select the hybrid cells. Now, to select the hybrid cells, there are different methods or techniques that are involved, like the biochemical method, or the visual method, or the cytometric method. These are the three methods that are involved for the selection of hybrid cells. So, coming to the biochemical method first. Now, biochemical method, as the name suggests, that the biochemical method, these are the compounds, which are the biochemical compounds in the medium, that helps to sort out the hybrid and the parent cells based on the differences in the expression of the character. So the hybrid cells are sorted out with, and they are based on the parent cells differences in the expression of the character. And there are two sub methods that are followed in biochemical method. The one is the drug sensitivity, and the other is auxotrophic mutants. So coming to the drug sensitivity first, that it is useful for the selection of hybrid of two plant species. If one of them is sensitive, that is the condition. That the hybrid selection is only possible if the two plant species are there, and then if one of them is sensitive to the drug. This is the condition. So the protoplast of Petunia hybrid can form microscopic callus on the MS medium, but are sensitive to actinomycin D. So the protoplast of Petunia hybrid, they form the microscopic callus on MS medium, but they should be sensitive to actinomycin D. It means that they should be inhibited by the actinomycin D. And the Petunia parodi, this is the second species. The first species we have is taken as the Petunia hybrid, and it is on a microscopic callus. They can form the microscopic callus on MS medium if they are sensitive to actinomycin D. But the second species that is taken is the Petunia parodi protoplast. So what the Petunia parodi protoplast does is, it forms the small colonies that are resistant to the actinomycin D. When these two species are fused, the fused protoplast derive both the characters. So when the species are fused, they derive the protoplast characters. And what it does is, it forms the formation of the microscopic colonies are formed, and the resistance to actinomycin D on MS medium is done. So it helps in the selection of hybrid by this way, that both the species are taken, that is Petunia hybrid and Petunia parodi, and then both the species, when they are fused, they derive both the characters. like the formation of the microscopic colonies is there, and also the resistance to actinomycin D on MS medium, which helps to the selection of hybrids. Now, the parental protoplast of both the species fails to grow, and the protoplast of the Petunia parodi form small colonies, while those of the Petunia hybrids are inhibited by actinomycin D. So this is about the drug sensitivity, that what the when the two species are taken and when they are fused together, how the selection of hybrid is done. So drug sensitivity, this technique was originally developed by Power et al. In 1976, and it was done for the selection of hybrid of Petunia, and also for some stomatic hybrids. This process was carried out. So the examples such as the hybrids between the Nicotiana sylvestris and also the Nicotiana nightingale, these two species were there, and the selection of hybrid was done. So the second method, in which is the auxotrophic mutants. Auxotrophic mutants, in that method, what happens is there are mutants which cannot grow on a minimal medium, and because they cannot grow on minimal medium, they require some specific compounds that are to be added to medium. So the nitrate reductase is deficient mutant of tobacco, and the parental protoplast of such species cannot grow with nitrate as the sole source of nitrogen, while the hybrids can grow through this process. So the two species of nitrate reductase deficiency, one due to the lack of apoenzyme and the other due to the lack of molybdenum cofactor, are known. So the parental protoplast cannot grow on nitrate medium, while the hybrid protoplast can grow on this medium. So the selection of auxotrophic mutants is possible only if the hybrid cells grow on the minimal medium. And there is also one limitation to the auxotrophic mutant technique, is that the insufficient or the small amount of the higher plant auxotrophs are there. So this is also limits this auxotropic mutant technique. Now, coming to the visual methods. The visual method selection of hybrid cells is efficient. Now, in some of the experiments, what happened that the chloroplast efficient protoplast of one parent was fused with green protoplast of another parent. So, what happened after the one parent chloroplast deficient was fused with the green protoplast of another parent? They facilitate the visual identification of the heterokaryon under the light microscope, and the heterokaryons are bigger and green in color, while the parental protoplast are either small or colorless. So this happened in the visual method that was carried out, and the identification of this heterokaryons has to be carried out to develop the specific hybrid plant. So there are two approaches in the direction, that growth on selection medium and mechanical isolation. So one is the visual selection, which is coupled with the differential media growth. Now there exist natural differences in sensitivity of protoplast to the nutrients of grown medium, and the media can selectively support the development of hybrids, but not the parental protoplast. So this is about the visual method. The mechanical isolation, the mechanical isolation is also the other method which is carried out in the visual method. So the visually identified heterokaryons under the microscope can be isolated by mechanical means, which involves the use of special pipette, namely Drummond pipette, and it is isolated heterokaryons can be cloned to finally produce some hybrid plants. Now, each type of hybrid cell requires special culture medium for its growth, and it can overcome by employing microdrop culture of single cells using pedal technique. So we have learned what is microdrop culture technique. You must be remembering that. Or if you are not remembering, you just go through that video and just learn that topic thoroughly. Then coming to the cytometric methods. Now, in the cytometric method, the flow symmetry and fluorescent cell sorting technique is there. And this flow cytometry or the fluorescent cell sorting technique, what it does, it analyzes the plant protoplast viability uh, so that the viability of cells can be maintained. So it is for the analysis of plant protoplast. And also same is applied for sorting and selection of the heterokaryon. So the hybrid cells derived from such selections are useful for the development of certain plants which are hybrid. So for the development of certain hybrid plants, this technique is carried out. So cytometric method is also carried out, which is with the help of flow cytometry and fluorescent cell sorting technique, which is done for the analysis of the plant protoplast and to maintain the viability of cell. And the same technique is used for the sorting and selection of the heterokaryon. So this is about the different methods for the selection of the hybrid cells. The methods are biochemical methods, visual method, and the cytometric method. Now in the coming lesson, we'll learn about the identification of the hybrid cells, that how the identification of hybrid cells is done. There are different techniques that are involved for the identification of hybrid cells. So if you like the video, don't forget to click on thumbs up button below, and also don't forget to subscribe for more video updates. Thank you for watching.